So with no further ado, let's bring him on. Andrew, welcome to the Biotech Voyager.
Wow, what an introduction. I can't believe it. The drum roll and everything.
You have no idea. That's a very long time coming. We've been trying to get the soundboard to work for weeks.
Yeah.
Ben's doing a good job.
I'm finally earning my keep. Andrew, how have you been? I know back when we did the Bio to Bedside, we had you on just to kind of have like a Y Combinator dedicated episode. Yep. And we had you guys on, and then also Leah Labs on. We had Leah Labs on last week, was it, Jeff, or the week before?
It's Velocity
They're spinning out a new
Therapeutics
company,
now.
So it's really cool to see.
Oh, that's so funny that we're like
yeah,
all on the same cadence. Amazing.
Mm-hmm.
Excellent. So yeah, bring us up to date since then. So before, you guys were in the very early stages trying to generate data, and now you've got the money to take this to the clinic. How have you been?
Got like a couple of detectives. The image that you used to like promo me coming on the show, I think is like the first findable image of Alex and I together ever.
Nice.
So that was fantastic. So yeah, back in 2024, we started the company. Based on work that was done at UCSF. And so this is work that Alex did during his postdoc. And what we were able to do is develop basically a very tumor-targeted vector that allows us to deliver genetic payloads. We're sort of calling this concept in vivo tumor engineering, right? So, in vivo CAR T, so hot right now. But what if you could think about approaches that could deliver the same outcomes for a solid tumor? So instead of modifying immune cells, you could go in and modify tumor cells. And instead of making them make, targeting ligands that you hope are present on the solid tumor, what if you could deliver therapeutic proteins? And so what we've been able to do is deliver a very potent IL-15 superagonist. And so that's sort of the first program that we're launching with. And then we spent the past 2 years, putting together the heart and soul of the company, putting together the core technology, starting to do development, and raising the capital that'll help us sort of get this first product into the clinic. And so now, it's great to be able to announce that, and we're really excited that hopefully early next year we'll be dosing patients and trying to make a difference in one of the most challenging solid tumors, which is glioblastoma.
Nice. Okay, so first,
yeah, people getting going into clinic. Yes.
Keep our studio audience under control, Ben. I have questions to ask. Jeez. So it's in glioblastoma. How are you getting the thing in there? That's the first question.
Yeah. So the great thing about working with neurosurgeons and neuro-oncologists is that they're great at delivery to tumors that are located within the CNS. And so for GBM, which is usually single locus disease, direct into tumoral injection works very well. And for basically the majority of viral therapies that have been delivered for GBM, they've all been local delivery. So we're using a catheter-based delivery system and going right after the tumor.
Nice. And then, so there are a few other companies who are doing kind of similar things. One of the ones that I'm thinking of is Siren Biotechnology. They're not doing IL-15. They're doing, I think, IL-12.
Yeah.
But what kind of vector are you using? Is it self moderating? I guess is kind of a dumb word to use, but you wouldn't want to have extended inflammation in the brain, right? So
yeah. So there's a couple of people who have thought about various versions of this approach, and the sort of goal is how can you try and get persistent therapeutic delivery in a tumor that's very inaccessible, right? So if you were trying to do like systemic delivery, has been very challenging in this indication. Because you have to cross the blood-brain barrier. So if you're going to be local, how do you get something to stay there, and how do you get it to be produced locally? Obviously, there's been an incredible amount of CNS development work for AAV. And so that's been the vector of choice for some other companies in the space. Trogenics is one. I think they're the one that's doing the IL-12. And then Siren is the other. And so I think there's obviously decision-making when it comes down to selecting your therapeutic payload. And some of that just comes down to how you think about translatability. And so for us selecting IL-15, what we thought was really interesting about that construct is it is cross-reactive in both rodents and humans. So when we're actually doing our preclinical studies, we're actually using the same exact construct that we're going to be using in patients. And so we think that for translatability, having that be a one-to-one understanding of how this vector is going to perform is much more helpful than having to design like a rodent construct and then hoping once you rejigger things inside of your vector that that's not going to have some subtle but maybe important impacts on what's going on in patients. And then it's just about thinking about the biology of what you think is going to be the best in terms of the signaling that you're trying to do with an inflammatory pathway. And so for us, we like IL-15 a lot. It's got a lot of really de-risked biology, and there's an approved IL-15 product in bladder cancer. So we think like taking the ensemble of data together, IL-15 just felt like a very good choice for us.
Yeah. So are you guys following along with Immunity Bio? The Anktiva is like kind of kicking butt.
Right. Yes, yes, very, very closely following that product, of course, and like hope for it to have continued success.
Exactly. Yeah. Good news for you guys.
Yeah.
Excellent. So I guess with regards to other approaches that have been on the market, right, for something like glioblastoma, why have other approaches failed here?
Yeah, so I think the thesis that we have for GBM and sort of more broadly for solid tumors has really come down to delivery. So as I was mentioning before, issues with getting therapy across the blood-brain barrier is really difficult. And so trying to solve this local production of therapy where it's most needed, which is basically in the site of the disease, we think that's one of the major barriers that we're trying to overcome. Beyond that, we're largely trying to flip cold tumors to hot tumors. And so trying to take this approach of being a little bit inflammatory, we think is very interesting and could be one of the unlocks for some of these difficult to treat solid tumors. There's been a couple of recent targeted therapies that have done quite well in DMG. And so there is, for people who possess those mutations, there's been some work moving forward. But when we're talking about really poorly mutated tumors that don't really have a wide variety of targets that are accessible in most patients, trying to go after something that's a little bit mutation agnostic, we think is also really interesting.
Yeah. So I'm wondering here, I'm a GBM guy, kind of. I did a postdoc in CAR T cells and GBM. And the big thing the biggest problem with that is like macrophage biology. They're like half the tumor is macrophages. So I'm curious if the choice of IL-15 had to do with like macrophage biology as well, or if it was just like, nope, super agonist the crap out of those things, get T cell activity, NK cell activity, and just like, shoot them with inflammation, basically.
Yeah, we were primarily looking at the immune side of things. And so when we were doing this study, and so it was just published in Nature Communications, which is the culmination of many years of work. It was great for Alex and the team at UCSF. And so we did a bunch of work trying to characterize the tumors post-administration of the treatment, looking for signs that were actually biologically following the sort of model that we're putting forward. Because in my opinion, right, rodent models, like the survival benefits that we're able to demonstrate are exceptional. But I think that rodent models have not really translated very well in terms of, curing cancer in a mouse doesn't necessarily result in a cure in human. But what I think mouse models are beneficial for doing is trying to validate that your biology is functioning in the way in which you're proposing. And so what we were trying to look for is signs that we're actually able to remodel the TME, and we are actually driving in T cell and NK cell populations. So through both flow cytometry and RNA-seq, you're able to show really strong evidence that that's occurring. So that's another piece of sort of building the foundation of why we think this therapy is so promising.
So I think I found the paper here. I'm going to read this thing later. So non-lytic viral immunotherapy induces long-term glioblastoma. Yeah, perfect.
Yeah, yeah. Put it right next to your bed. It'll help you. You'll do a great
Put
job.
it under my pillow.
Sleep will never do better. Yeah.
Yes. Perfect.
the biotech fairy comes along and leaves you $5.
Yeah. And your IND approval under your pillow.
Exactly. Yeah, I found that like passive diffusion is generally the best way to ingest scientific knowledge. Yeah.
Yes,
exactly. Yes, from paper to brain.
Yes.
Excellent. Okay, so I'd be curious, Andrew, just to go back, and folks, for those who are watching again, thank you for coming. If you have any questions for Andrew, feel free to put them in the chat, and we will get to them post-haste. But just, I guess, for you, Andrew, again, this is something I'm really curious to double-click on, which is, again, you took an approach that I'm very interested in, right? In that you went the Y Combinator approach, which hasn't been super common inside of kind of the wider biotech space. Why was that, and kind of where do you see this going in terms of like Y Combinator's interest in biotech in the future?
Yeah, for sure. So I think we're in like a sort of Cambrian explosion moment for founder-led biotech because the cost structure is just completely flipped. So I think like much in the same way for tech that like the advent of like the cloud storage like AWS and Google Cloud, et cetera, made it easy for basically anyone to spin up a tech company. I think now that the cost of getting to clinical data has gone down by a factor of 10x, I think that'll just enable a lot more founder-led biotech companies to get to an inflection point where they can then raise capital in terms that are more similar to a tech company. And so I think the reason why this path was not common in the past is because perhaps you were reliant on like a large incumbent fund to sort of escort you through the early stages because the requirements were so high. But in the current environment, again, with the cash that we've been able to raise, we'll be able to get human data. And I think that's about probably like 50 to 75% less than it would cost, even 5 years ago, what people were thinking about. And this is like a cell and gene therapy. This is a very complicated biological product. And so I think that when those cost structures flip, these models become more attractive. And so I think for founders who would like to be in the driver's seat for their companies, going through the Y Combinator route is a good way to do that. And I think the network supports that. And you can get enough capital now to get to these inflection points where you can really make a difference.
That I love to hear that because this is a little bit of a self plug, but we are starting this platform called the Biotech Voyager that's going to be explicitly dedicated to tracking these early stage companies. And I think like I really like the tech industry where it's like everyone and their mother on Twitter has got their own tech company. I'd love to see that in biotech where it's just like we have all these really new, young, gritty, like millennial-y kind of Gen X biotech companies that are like some guy. Ben, you are the one kind of blowing this horn 6 months ago. Some guy in his basement with an AI program designs some therapeutic, gives it to his dog.
Yeah, the dog story.
Right?
Yeah.
And it's like,
it's crazy.
Okay, I might be a little bit more on board with this like new agey like biotech synthesis.
Yeah. And the other story that's been really interesting to follow is Sid from GitHub.
Yes, GitLab.
Where he basically really aggressively took on ownership of his cancer care, went and developed a bunch of personalized treatments in a way that most people wouldn't have thought was possible, 10 years ago because of the tools that are available now.
Yeah. It could be
pros are coming for biotech. I've been telling you.
Yeah, a part of me is like not envying the FDA. Because it's going to be a regulatory nightmare. I think it's going to be like we're going to have so many. It's cool on one hand, but immediately like the biologist in me is like that's dangerous. So what are your thoughts on that?
So I mean, 100%. So I think that doing this in sort of a risk appropriate manner is going to be the path forward. And so the way that I think we could start to think about things is look at what people are doing elsewhere, right? So we don't have to reinvent the wheel. We're not it's not like there's no biotech activity going on outside of the US. So we've actually been able to benefit from sort of surveilling what people are doing in other jurisdictions and seeing what works and being able to learn from what doesn't. And so places that I think are good examples of how you can do this in sort of a risk appropriate fashion would be Australia, right? Australia is a great example. That's been a pathway that people have been doing for probably now the past 10 to 15 years. And they just have a very different approach to early phase clinical studies than the US does, where again, patient numbers are on the smaller side. And so if you're thinking about, the risk of a cardiovascular drug that's going to be in 2 million people, like, once you're making a decision on whether or not that level of exposure to the population is going to occur, I think you need to be very risk averse. But when you're dealing with, serious life-threatening diseases in, 1 to 10 patients, I think you should sort of right size the requirements to get to that stage to see if this is worth pursuing further. Because if it's not, you can just think of the hundreds of millions to billions of dollars that go into programs that ultimately fail relatively early in clinical development. And so the less expensive we can make those failures, the more potential successes we can have. And so beyond Australia, obviously the other example that now everyone's doing is China. And it's very similar in the way that they have sort of decentralized the review process for early stage work. And again, I think sort of taken a risk benefit framework to small early proof of concept that I think is really interesting for trying to sort of drive through biotech innovation in a way where it hopefully no longer costs us like $2 billion per drug to approval.
Yeah, and I'm thinking that this is a little bit of a long shot and might sound like completely stupid. I don't know. Here we go. I think that with the maturation of AI-based analysis, we might be able to get to a point where we can like have relatively confident and robust like benchmarking very, very, very early to say like, here's the signatures that we're looking for in a pre-IND therapeutic that will predict phase 2 efficacy. Maybe not now, maybe not in 5 years, but maybe like 10 years, we'll be able to get like enough data that was generated with the purpose that says like, we can now be predicting things very, very, very early and maybe cut things off before we have to figure them out in the real world, quote unquote.
Totally. Yeah. Part of me is very optimistic for that. Part of me also wonders how many potentially good drugs were canned at the rodent stage.
Sure.
Because the rodent biology is not representative, and they were just like, "Oops, we'll never touch that again." But it's just like maybe there was something there that we.
I
don't imagine.
There are some companies. We featured one, I think last week, where people are kind of like buying the shelved drugs from pharma that may have had like maybe lackluster phase 1s or phase 2s. And they're now saying like, let's give that a relook. So that's kind of happening, but I think, yes, like just like scouring over all of the negative results from like, years past could get really interesting.
Yeah, I think my like axe to grind is I think that sometimes we get really infatuated with like large numbers and screens. Like a lot of times people get really excited. We did 500 million variants. We did 1 billion variants. And like what actually matters is the space that you're sampling. And so like looking at very high signal areas with smaller things. So like looking at drugs that have already had human experience and trying to find where they might fit is probably a much more efficient approach to getting winners than trying to sort of boil the ocean when there's really only like 2 or 3 grains of sand that are interesting, right?
Sure. Yeah, I think that when I was in grad school, they had a whole initiative of like drug repurposing, where they would do just that, where they would take the repertoire of approved therapeutics and try and see like, hey, does this heart drug work in glioblastoma, right? Or like, does this oncology drug work in heart disease, right? So there's also, I think too that the massive fishing experiments might not be as difficult to arrive at a conclusion because of AI these days as well. But in the past, it's just kind of like we can just take all the things that our chemists can produce, throw it in these cell lines, and see if anything sticks.
We have robots now. Let's just run, 96
35 million.
Yeah.
Right.
All right. So there are a couple comments. Just want to get through here quickly. Rob, you might have your question cut off here. Might be a silly question for you, Andrew, but is turning a GBM tumor hot? I'm not sure if that question got cut off.
So it's like immunologically going from a cold tumor to a hot tumor. So like, yes, that's what we're trying to do. So GBM is traditionally thought of as a cold tumor, as our most solid tumors, in that their microenvironment is basically immunoexclusionary. So if you were to like take a biopsy and look at all the cells and see which ones are immune cells versus which ones are tumor cells, you'd find like only very, very, very few T cells there. And so what we're trying to do with the cytokine that we put on board is to dramatically increase the presence of those immune cells within the tumor, and then drive them to start killing these aberrantly mutated cells that they encounter.
Yeah, it's a very strange term. It's immunologically hot, right? People think, oh my gosh, you don't want to turn it like the temperature up, right? In your brain.
I just think of this all the old cards. GBM so hot right now.
Exactly. Yeah.
Chris Pivey also has a comment. Sorry, wrong one here. Oops. Here we go. Chris. Yes, patient stratification also falls in the same bucket. I think he's going back to just like the cost of.
Yeah, and I think knowing the biology better is one of the things. The first time that I really appreciated it was with Avenzo Therapeutics. The key to your phase 1 and phase 2 success is patient stratification, especially phase 2. Knowing how your drug works and knowing which patients it won't work in is one of the most important things. But it requires you to know how your drug works, which is usually like not as well thought through as you'd hope.
Yeah, well, I think it was, what is it, like the Keytruda versus Opdivo, like one of them stratified better in terms of PD-L1 expression, and that's why Keytruda won.
Yeah, makes
and
that,
so yeah,
that sounds...
it can come down to these early understandings of mechanism and trying to make sure that you're trying to enrich your population for responders, and then you can get more and more understanding as you get to a larger and larger data set.
Yeah. The Avenzo Therapeutics one was they have a CDK2 inhibitor and they stratified for high cyclin E expression, which would make it sensitive to CDK2 inhibition. So it's like knowing how your drug works, finding the patients, getting the right biomarker, and that will be easier with our increased ability to do like tumor biopsy based approach, like analysis and AI, things like single cell and like high throughput IHCs. And then knowing how your drug works is like the other half of that. So
totally.
Okay, Rob's question did get a bit cut off, and it was just with he was going to ask: are there risks in terms of like the pressure or inflammation of a productive immune response in the brain? Oh, that's a good question.
Yeah, so this is a really good question, and when you're thinking about these treatments, right? You want to induce an inflammatory response because that's almost a signal of potential efficacy. But then, of course, within the CNS, there's potential risk to that. And so when designing the clinical protocol for these types of drugs, you always include sort of an inflammatory control schedule, where if there are signs of clinically risky inflammation occurring, there are a number of things that you can do to tamp it back down. And so, as a part of any reasonable clinical development plan and any reasonable clinical trial, you think about these potential, on-target, off-target effects of your drug, and then you think about basically in trying to design control mechanisms to maintain the safety for the patient.
Love it.
Yeah, Siren Biotechnology, they have like an AAV based one. So it's like as the cancer cells die, you're going to get less expression of interferon beta. That's like, there's got to be some kind of like safety system built into the therapeutic, or you're just going to get, brain damage.
Right. But yeah, you can dose with things like steroids and other things to tamp back inflation.
True. Yeah.
Super cool. Okay, so I guess just in terms of in terms of like the data that you've been able to generate so far, like what would you say was the really compelling piece that I guess made prompted this investment to go forward into the clinic?
Yeah, so the thing that first caught my eye, and I think has been largely the driver of people's belief in the technology, has just been the efficacy signals in multiple really aggressive GBM models in rodents. And so these are very, very fast growing. They grow substantially faster than what you would see in a human. And so if you're able to control those tumors to the point where we get basically complete regressions in some models, that's a really strong signal that you're making a big difference. So I think that's one. People like survival benefit, and that's sort of a good metric. And then the second one for me, which I think is most important, because I'm always just thinking about the human biology is going to actually rule the day. Like we don't get approved based on rodent data. So it's how thoroughly have you vetted the way that your therapy works, and how much have you validated that it's doing what you say it's going to do. And so for us, really looking at the detailed mechanistics of like, does the virus selectively infect the tumor? Does it replicate within the tumor? Does it produce functional payload? Does that functional payload induce the correct biological response? And the fact that we were able to validate every step of that pathway in exquisite detail was for me what made me want to take the leap and go and like, let's actually push this forward.
So I have such a nerdy question for you.
Yes.
It has to do with the data from the paper. I'm looking at some of these of like the mouse data. I see TU2449 rechallenge data.
Yes.
So when I see rechallenge, normally I think of like things like vaccines. When you guys do the rechallenge, do you put the tumor in the same spot? That's the first question. And do you perfect? Okay, good. Yeah. And do you give the drug again, or is it like
we do not. We do not. Yeah. So this is like yeah. So again, as a way of sort of validating that we're doing an immunologic mechanism and not some virus response that's being triggered, locally or whatever. That was why we did the rechallenge experiment. And so seeing tumor rejection, I think it was like 65 days after the first tumor, shows that you're really creating like durable immune memory within the immune cell population of the animal.
Yeah, and the big thing with GBM is like recurrence. You can,
correct.
It's like almost you need to be able to, you need to be able to have your thing have an abscopal effect, or you're done. Like you're gonna have metastasis in the brain and recurrence. So you've got to be able to rechallenge and get a good response in different locations. So really good to see that.
Yeah,
it's
that durability. Yeah.
Yeah, and it's a straight line, 100%, no mouse deaths to report. Which again, we can cure cancer in a mouse. We've done it over and over again. But getting it into humans, but it's I mean, what else do you do, right? You've got to
it's the best. Yeah, you work with the tools. Yeah, you work with the tools that you have. And again, for me, it's about answering mechanistic questions, and like survival is a sexy figure, and like great, we have incredible survival data, like so good for people who care about that. But what I care about is like, is it doing the biological things that we say it's going to do?
Yeah, do we see like CD8s and like antigen specific?
Exactly.
Yeah,
exactly.
Nice.
Exactly.
Yeah, no, it looks really nice. I'm gonna dig into this later, and maybe I'll have some more questions to ask Alex.
Yeah, it was very strategic for me to not share it before we came on.
Nice.
Yeah, get the friendly questions down. I'll get the hard ones later.
Great. Yeah. No, I'll be nice. I promise.
Yeah, so I guess just in terms of next steps here for the company. So I think I said right, you're planning on doing your clinical testing in China.
Yes.
I would be curious to, like, first of all, we can jump into that, like what are next directions for the company, but I would be curious to just kind of jump into that as well, right? With like, especially for someone in your position, right? You mentioned earlier, right, the cost of developing these therapies and like evaluation and whatnot has reduced by tenfold. I'd be curious to get your thoughts as well on just like what role China is playing in all this and kind of where you see them slotting in the future. There's lots of debate going on right now on biotech Twitter about, whether or not this is kind of like spelling the end of US biotech dominance, or if there's going to be, different parts of the industry just getting outsourced to different parts of the world. I guess maybe let's jump into that, and then we can end off with directions of where you guys are planning on going next. But I'd be curious to get your thoughts as someone who's in the weeds of this right now.
Yeah, so I mean, I think still innovation is coming from the US, and you're seeing the most novel technologies coming out of the US. Funding for that high risk research has been dramatically in flux, I would say, for the past couple of years. And so if that funding is not there, I do think we will see dominance in innovation. And so that'd be the first thing, is that if you don't invest in innovation locally, you won't be able to maintain it. And the people that are innovative are some of the most mobile people on the planet. And so they will go where there is capitals to support them. So I think if we want to support innovation, we need to invest in it locally. I think that the development side of things is and should be just kind of like an open marketplace. Like these are in many ways commodity services. And so we should be able to go to the lowest cost provider, because once again, that'll allow us to try to develop more drugs. And the more drugs we put into the pipeline, the more successful drugs we get at the other side. I think that once you get to approved drugs, then it again becomes, I think, interesting and important to invest in domestic production and to support that. Because I think these are like critical pieces of the economy and the healthcare infrastructure. And so you would want to be able to not be reliant necessarily on, other parties for all of them or for all the raw materials. And so like, that's where I think I land on, is like, you need to support innovation. I don't think there's a better like cultural tolerance for risk anywhere else than the US.
Yeah.
But then I think once it gets to development, that should just be a commodity, and we should want to push the prices of those tasks as low as possible, because then it gives us better benefit for approved drugs. And then, finally, once it gets to approval time, we should want to have a robust manufacturing infrastructure for late stage and commercial products, so that we can support them at home and abroad. So that's where I think about that. And then in terms of how we think about the context of doing drug development in that world, again, our job is to, as quickly and as cost effectively as possible, understand the human performance of our technology. And so that's the mission that I'm on right now. And then as soon as I have that understanding, obviously the US is going to be where we're doing the majority of our development work for phase 1 to phase 3. And so, right now it's about taking technical risk off the table. And then it's about, doing the best sort of commercial and, where's the best clinical trial infrastructure in the world for mid to late stage development.
Yeah, that all makes perfect sense. Yeah.
Yeah, and it's crazy. I don't know why it's like such a like, oh my God, we have to scream at each other about this. It's like, no, let's just kind of all be reasonable.
Yeah, and so the one thing that I might want to push back a little bit on is like, if America is the one that's fueling the innovation, shouldn't it be the people in America who potentially benefit from like the clinical trial participation? You know,
I mean, I think they do, right? The drugs that are developed through this process are first available in the United States usually, right?
So if you do a clinical trial, if you do your phase 1 and phase 2 in China, though,
yeah,
then like the patients in America aren't eligible for that.
I mean, that's like 200 patients, right?
Okay,
so
that's almost,
yeah. So if like, and like it's 200 patients in an unproven drug. So I think that what I, you know, the people that sign consent waivers and actually participate in clinical research are incredibly brave and are like hugely important for society. But I don't think that we should think about those patients as being like the access strategy that we care about, right? The access strategy we care about is where do approved medicines launch first. And that's in the US. And so the second point beyond that, right? So like who's going to be the first 10,000 people to get access? They're probably going to be in the US. The second question is then what happens when those drugs go off patent and then more and more people in the US and worldwide are able to do it. And so I think that it's a system, again, that rewards risk-taking for breakthrough medicines. I think there are potentially bad actors that are trying to leverage some of the economics in ways that are, you know, financially beneficial and not really participating in the innovation part of the business. And we should probably think about how to fix that. But I think in order to reward the risk and just facts of how many drugs fail along the way, and you need to be able to support those on the back end. I think we need a system that is able to financially support that, and also is still giving, you know, U.S. patients the first access to some of the best medications that are coming through. Yeah.
And the thing about the you know, the clinical trial patients don't benefit in the U.S. It's like, you're right. It's like 50 people. So shut up. We're gonna get this thing, and we're gonna prove it. We're gonna prove it, and then once we know it's good, we can offer it widely.
Exactly. And like the faster we get through trials, the more people will be able to do. So like if I'm stuck trying to enroll a 200 patients study in the US and enrollment is going slow, that seems way more problematic for potential patients that would benefit than if I got approved 2 years sooner and opened the floodgates to the masses 2 years sooner.
Exactly.
And so yeah.
Clip that. That's yeah. Perfect.
I don't know, you just have to take a step back from like the, you know, screaming and yelling that occurs on the internet, and then just try and like go to first principles and think about like, what is the world that you want to live in and how do we make that happen, right? So just
exactly.
Put those pieces together. Yeah.
Gotcha. Okay, we have a second question here from Chris Spivey. We love Chris over here at the Biotech Voyager. Shout out, Chris. Andrew, when first broaching your approach to new contacts, what is their most skeptical question or most difficult aspect for them to grasp? Chris coming in with some hot questions.
It's a good question. I think generally gene therapy is harder for the layperson to grok and understand. And so like getting people to think about like how it works, we usually spend a lot of time talking about how it works and just like what are the steps that occur. Because, you know, chemotherapy, antibodies, they're sort of just like very understandable, like lock and key kind of stuff. People can understand that pretty quickly. It's been around for, you know, 100 years for drugs and, you know, pushing 50, 75 for biologics. And so it's just for gene therapy, when you have to talk about sort of all of these multi-step processes that occur, getting people to understand.
Well, you see this thing called transcription and gene expression.
Yeah, you're like the mitochondria is the powerhouse of the cell. It's like, you know, you just like have to.
my wife's one biology fact that she knows what the mitochondria is, and I'm like, but there's this like, you know, RNA polymerase 3 does this, and it's like,
right. And so and so like how up and down the stack can you go with people to get them to understand what are the necessary and sufficient things that are happening to make your drug work. And so, having that conversation is usually where things start. And once we can get them across the sort of value of understanding on that, then it comes into like, how fast can we get this to patients? And
I'm sure that the mRNA based like COVID vaccine didn't really help with public sentiment. There's probably a lot of mixed feelings about like, oh, it's like the COVID vaccine. It's like, well, not really, but okay.
Right. Yeah, I mean, you can't control what things end up like bubbling up to the public consciousness.
Right.
And so then you just try, we like to use I Am Legend as a sort of touchstone, right?
Okay. With Will Smith, that one.
Yeah. Well, and it was a cancer vaccine that originally created the zombies.
Oh, really?
That was great. Yeah, it's great.
Nice.
It's great.
It seems to be mildly in line with my gripes over cancer vaccines, i.e., they'll never work to treat cancer. They might do something else, but it's not going to treat cancer.
Yeah.
All right, Andrew, I know we're a little bit over time. We appreciate you coming
Yes.
on. We do ask, there's one question, just we try to get like a pithy take from you, 30 seconds or less.
Oh, for sure.
And the question is, what's slept on right now in biotech that people should be paying attention to?
I think China is still slept on. I think like as much hype as you just have to go there. Like everybody who works in this space should just go there, like see it on the ground, interact with the community. I think that US companies have just assumed that they would be dominant and the fastest option to do things. And it's like going to Shenzhen for electronics. Like you just need to kind of see it to understand what we need to be able to do if we want to be competitive here. Yeah.
Excellent. On that note, thank you so much, Andrew, for coming on. We know we're a little bit over.
Yep.
Appreciate you moving around your calendar. We're looking forward to following you guys into the future. Cheers.
Yep. Talk soon, guys. Bye-bye.
Thanks, man.
If you like this video, subscribe for more. We've got a boatload of content coming. Also, who should we talk to next? Drop your suggestions down below.